Inside a developing tumor, the immune system often constructs ectopic outposts known as tertiary lymphoid structures, or TLSs. These localized clusters of immune cells assemble directly within the malignant tissue to coordinate anti-tumor immunity. Identifying these structures currently relies on routine pathology. A technician prepares a standard biopsy slide and scans the tissue under a microscope. The resulting clinical assessment follows a binary protocol. The pathologist evaluates the sample and assigns one of two labels, TLS present or TLS absent. This chart shows the limitations of that binary label. These two patient profiles are both classified as TLS detected, yet their outcomes follow drastically diverging survival curves. Relying on a simple detection metric fails to consistently predict therapy response across different cancer cohorts. Treating these structures as monolithic entities ignores the underlying biological complexity that dictates clinical outcomes. To address this inconsistency, researchers conducted a pan-cancer analysis mapping high-resolution spatial transcriptomics across 340 tumor samples and 12 different cancer types. Instead of just searching for the presence of immune cells, the study analyzed the precise maturation state and cellular organization of every individual structure identified. The team mapped the physical coordinates of these formations relative to tumor cells and measured the specific signaling gradients radiating through the surrounding malignant tissue. These researchers then developed a pipeline to translate this complex spatial data into a scoring system that an AI classifier can apply to standard pathology slides. Shifting to a quantitative multidimensional model captures the prognostic information within the immune microenvironment that a binary label misses. Tertiary lymphoid structures are dynamic ecosystems. They begin to assemble as immune cells migrate and clump together to form organized clusters within the tumor bed. The first stage is the unorganized early TLS. In the primary stage, the cluster develops defined zones and follicular dendritic cells. The secondary TLS is fully mature, featuring active germinal centers. Pan-cancer data shows 55% early, 31% primary, and 15% secondary. These stages also exhibit intratumoral heterogeneity. A single tumor rarely contains just one type of structure. It typically hosts a mix of all three maturation stages simultaneously. Labeling a tumor as TLS positive is inaccurate when more than half of those structures are too immature to function as advanced immune hubs. Beyond internal composition, a structure's physical location geographically impacts surrounding tumor architecture. In the proximal zone, closest to the TLS, we see heightened immune activation and antigen presentation. Conversely, the distal zone is dominated by oncogenic proliferation and aggressive invasion programs. Researchers isolated these opposing forces using spatial transcriptomics, which allows for the mapping of gene expression data directly to physical tissue coordinates. These distinct signaling gradients were consistently observed across multiple tumor types. A TLS acts as a biological anchor. Its physical location dictates a topographical gradient that dictates local tumor behavior. Translating these complex spatial biology insights into routine clinical workflows requires moving past expensive and highly technical, low-throughput laboratory techniques. While spatial transcriptomics provides high-resolution genomic mapping of tumor tissue, the technology remains expensive and technically complex for routine use in hospitals. To bridge this gap, researchers trained an artificial intelligence model to extract maturation data using standard H&E stained tissue slides. The model analyzed over 25,000 structures, cross-referencing genomic data to learn subtle morphological shapes visible on routine pathology slides. The resulting AI can automatically segment a standard tissue slide and color-code the specific early, primary, and secondary maturation regions. The model achieved high diagnostic accuracy and successfully generalized to cancer types that were not included in its initial training data. This pipeline connects high-dimensional biological discovery to scalable clinical application. Using this AI classifier, clinicians can generate a maturation-aware composite score for a patient using routine pathology samples. This score is a mathematical calculation based on the weighted balance of early, primary, and secondary structures found within a single tumor. This series of Kaplan-Meier graphs shows the prognostic value of this approach. The composite score creates mathematically significant separations in patient survival, providing clarity that simple detection metrics cannot. This prognostic power remained significant regardless of tumor stage, and accurately predicted patient responses across immunotherapy and chemotherapy regimens. However, the data also revealed a nuance. Fully mature secondary structures are not always beneficial. As they reach peak maturation, some structures develop regulatory programs and T-cell exhaustion signatures, acting as immunoregulatory niches rather than pure attack hubs. Predicting patient outcomes requires measuring this balance of maturation and exhaustion, rather than searching for a single dominant structure. While these spatial maps reveal how the tumor microenvironment is organized, we are still uncovering the mechanisms that control this physical growth. This research is observational. It establishes clear correlations between maturation and survival, but it does not yet provide definitive proof of causality. We do not yet know if mature structures actively drive tumor suppression, or if they form as a byproduct of an immune response that was already effective. The next stage of research involves functional perturbation, artificially manipulating TLS formation to observe the direct effect on tumor progression. Replacing the binary label with a quantitative, spatially resolved framework establishes the baseline required to engineer more precise predictive clinical biomarkers.